Chapter 15, vocab video. The first word is morphology. Morphology literally means the study of shape of something. So when we're talking about using morphology as a diagnostic technique, we're talking about taking a sample from someone and looking at it under a microscope. Are there bacteria there? Are there fungi there? Are they rods or cocci? So just looking and studying the shape using a microscope. A dichotomous key is sort of a flow chart. You actually made one of these, a simple one, in one of your lab activities. But it's basically just a flow chart that helps you get to a diagnosis. So, the first thing you might do is gram stain. So then, is the bacteria gram positive or gram negative? If it's gram negative, you follow this part of the flow chart. All right, is it a coccus or a rod? And then follow this part of the flow chart. So it's just a key and a guide to help narrow down different types of bacteria based on results of testing. Polymerase chain reaction, we've talked about before, is a really powerful technique to amplify DNA or to make many, many copies of a piece of DNA. It can also be used to look for specific types of DNA to test for specific organisms. So they can do PCRs to look for HIV or flu, and if you get a positive result, because you can make lots of copies, that means that you do have some HIV DNA in your sample, and if not, if there's no HIV DNA in there, and then you get a negative result. So PCR can be used in biotechnology and genetic engineering to make copies of genes that you want to clone, but it can also be used as a diagnostic to look for the DNA of certain pathogens in like a swab or a blood test. Hybridization is a technique that can be used, again, using we're looking for DNA here, so you use these DNA probes. So you get some, basically you can create little pieces of DNA that are fluorescently tagged, and they are complementary to viral DNA or bacterial DNA specific sequences. And then you can take the patient's sample and you blot that sample onto a piece of filter paper, and then you add your fluorescent DNA probe. And if the fluorescent DNA probe sticks because it found its matching DNA because you're infected with that virus, then that's a positive result. If it doesn't stick, it'll wash away if there's no matching DNA there. So hybridization is an assay to look for DNA of a pathogen, and it usually uses some kind of fluorescent or colorful indicator that indicates whether it's positive or negative. Serology is the study of your serum, which is the portion of your blood that contains antibodies. So basically, the serology, there's a ton of different types of serological tests. They all involve looking for antibodies. An agglutination test is another test that is a type of serology test. It uses antibodies to test for certain antigens. So a common agglutination test is done when blood typing. So when you type blood, you put a drop of blood in with a drop of basically some antibodies that match the different antigens. So if you have anti-A antibodies, anti-B antibodies, when you add the right antibodies, the antibodies will bind to the blood cells and clump them all together. And so if they start clumping or agglutinating, then you know that that is the type of blood that it is. So a Western blot is an assay to look at proteins. Basically, you run these proteins on a gel. So you have like a slab of Jell-O with some little wells in it, some little holes, and you pipette or squirt your protein sample in those wells, and then you run an electric current through this gel, and the proteins will move through the gel and make these different bands, and then you can see if that banding pattern matches specific pathogens. Also, you can use antibodies to probe that gel to see if any of those proteins in it match the antibody that's made towards a specific pathogen. So it is a process of running proteins on a gel and probing it with antibodies. Titer is a measure of antibodies in the blood. So after somebody has a vaccine, is vaccinated, they may have a titer drawn, they may have blood drawn to calculate or quantify how many antibodies are in their blood in order to assess whether that vaccine was actually effective or whether they're in need of another booster shot. An ELISA, that's an acronym, ELISA. It stands for Enzyme-Linked Immunosorbent Assay. This is another type of serology test where you use antibodies to detect an antigen, or you can use antigen to detect an antibody. It's a common test that's done for testing for HIV, actually, because it's very sensitive. Any type of test that uses antibodies is very sensitive, meaning it can detect even very small amounts of antigen. The tuberculin reaction. This is the tuberculosis test. It's also called the Manteau test. It's a little shot of tuberculin protein under the skin subcutaneously, and then you come back to the office a couple days later and they assess how much swelling there is. So you have a hypersensitive allergic reaction basically if you've been exposed to tuberculosis. If you're positive for tuberculosis, you'll have a hypersensitive reaction to that tuberculin protein. It's also called a PPD test, purified something, protein derivative, I think, because they're just injecting this tuberculin protein in your arm and looking for a swelling reaction. A microarray is an assay to detect little pieces of DNA. So it's a genetic test, and the neat thing about it is you can put lots of different DNA probes on a tiny little like microchip, like plastic chip. So you could test for lots of different sequences at one time. So you could potentially test someone for 100 different viruses at once, just looking for DNA from those viruses. So it's a quicker way to diagnose, but it's not quite cheap enough to be used sort of, regularly in hospital labs, but in specialized labs and definitely in research it's used. And then the last technique is called mass spectrometry, and this is a big fancy chemistry machine that breaks up proteins into little pieces, and then someone who's trained to read the output can figure out what that original protein was. So it's a way to identify proteins using like a machine that chemically blasts them apart. So it does not involve genetics, and it does not involve antibodies. It just involves taking a protein sample, putting it in a machine that tells you what the protein is. That's very oversimplified, but it's as much as I understand about it. So, chapter 15 for you.